This is Module 5 about translation. In this video, I'll be discussing the genetic code, which is the codon code used by ribosomes in order to create a polypeptide structure using RNA as a template. The template for translation is the messenger RNA. Within the messenger RNA, this is where you find the information to encode for proteins, and ribosomes are the enzymes that decode the message within the messenger RNAs. The structure and function of proteins are that proteins are long polymers of amino acids linked together by peptide bonds, and peptide bonds are formed with the help of the ribosome. The amino acid sequence is the primary structure, and this is the structure that is created by the ribosome. Once it is created, that polypeptide structure will fold to create secondary and tertiary structures, more and more complex levels of protein folding. And then if two or more polypeptide chains associate together, this forms a fourth level of protein called quaternary structure. This is a list of all of the biological amino acids in our bodies. And so here is a standard baseline amino acid, and these are the groups that are common to all 20 amino acids listed in this chart. And so it has a central carbon linked to an amino group, a carboxyl group, a hydrogen, and some form of R group that sometimes has a charge, sometimes does not. Sometimes it's an aromatic group, sometimes it's nonpolar, sometimes it's positively charged, sometimes it's negatively charged. And the nature of the R group, or the chemical nature of the R group, is what directs the protein to fold properly. As I mentioned before, amino acids are joined together by the peptide bond, which is a condensation reaction also called the dehydration synthesis reaction, in which the carboxyl terminus of one group becomes covalently attached to the amine terminus of another group to form the peptide bond. Again, peptide bonds are what holds different amino acids together in the primary structure, and this is the structure that the ribosome is responsible for creating using RNA as a template. And then once the primary structure is created, the secondary, tertiary, and quaternary structures will form based on interactions between the R groups and also interactions across the peptide bond. If you're really curious about how proteins fold, don't worry. You'll get a lot more of this when you take biochemistry in your third year. Okay, and so how does the ribosome know which amino acid to link and form a peptide bond in order to create the polypeptide chain? And how does it know what order to put them in? Well, the ribosome is decoding the messenger RNA, and it does so based on sets of 3, which are called codons. So the genetic code consists of 64 different codons that encode for the 20 different amino acids that we see. So for example, 3 U's together signify in the mRNA signifies to the ribosome that a phenylalanine amino acid should be placed in at that position. 3 C's together signals to the ribosome that a proline should be placed in that position, and so on. And so to read this chart, if you are faced with a codon like you have GAU and you want to find it in your genetic code, GAU starts with the first base of G, and so all of these start with the first base of G. GAU is the second base is A, and so you want to go where G and the second base A converge, and so you are restricted to this quadrant here. And then GAU is right here, and it encodes for aspartic acid. The codons here are written in the 5' to 3' direction as they appear in the messenger RNA. And so if you wanted to know what, say, AUG is in the template DNA, it's AUG here in the 3' to 5' direction in the RNA, and so it would be TAC in the 3' to 5' direction in the DNA. Remember, RNA is complementary and anti-parallel to the DNA template. So you might have noticed something. We have 64 codons. Oh, I should mention, we have 64 codons. 61 of them are what's called sense codons, meaning they encode for something. They encode for some amino acid. 3 of them are nonsense codons, meaning they don't insert any amino acid, but they do tell information to the ribosome to stop translating this protein. If we have 64 codons, but only 20 amino acids, That means what we actually have are, what this represents is the degeneracy of the genetic code, meaning an amino acid may be specified by more than one codon. So for example, let's look at lysine, LYS. Lysine is encoded by these two different codons. And so two codons both encode for lysine. Let's look at leucine. Leucine is LUE. Leucine is represented by 6 different codons in the genetic code. And so all 6 of these codons will encode for the same amino acid, leucine. Synonymous codons are codons that specify the same amino acids. And so everything in this box, these would all be synonymous codons. All of them specify leucine. ISO-accepting tRNAs are different tRNAs that accept the same amino acid but have different codons. And so the tRNA that recognizes CUG is different from the tRNA that recognizes CUU, but both of those tRNAs are linked to a leucine. So these would be ISO-accepting tRNAs. So this 64 codons for 20 amino acids introduces a lot of degeneracy within the genetic code. There's also what's called the wobble hypothesis, where a single anticodon in the tRNA can pair with more than one codon in the RNA. And so about 30 to 50 tRNAs can actually pair with about 61 sense codons. Let's see what this actually looks like. Here we have a tRNA that has a single anticodon, AGG, that binds to and recognizes the codon UCC. So this is a normal Watson and Crick base pairing. And this tRNA is linked to a serine. And so UCC in the RNA encodes for serine. At the third position, the codon, the pairing is actually relaxed. And so UCU will also sometimes result in a serine, because this position in the third position, this G in the third position, doesn't always have to follow normal Watson and Crick base pairing rules. These pairing rules are relaxed at the third position of the codon. And so while this tRNA has one codon, AGG, it's able to make a nice bond with this UCC, or association with this UCC, but it can also pair with UCU, which is not typically supposed to encode for serine. UCU, oh no, I'm just kidding, it doesn't encode for serine, I'm sorry. So both of these, UCU and UCC is the one. Here's UCC. UC, third base is C. UCC also encodes for serine. And so this is just illustrating the point that two codons, UCC and UCU, can both encode for serine, even though they have different, even though there's different codon in the RNA. It also, this one codon in the RNA will bind to one type of anticodon, even though it should bind to two different types of anticodons. And so this one tRNA is supposed to recognize this codon, but in fact recognizes both of these codons. And so both would encode for serine. Okay, the RNA is translated into proteins using something called the reading frame. A reading frame is the set of 3 codons per amino acid and how the ribosome reads it. And so it's really best to explain what a reading frame is using an example. Here we have one RNA template reading 5' to 3', and proteins are created from an RNA template in the 5' to 3' direction. And so the ribosome moves in this direction down the mRNA and creates a polypeptide sequence, with the N terminus coming out first and the C terminus coming out last. These codons are non-overlapping, and so if the ribosome starts reading AGU as a single codon, it will shift down 3 bases and then read CUU, and then read ACC, and then read CGA. This is one type of reading frame. Now we can take this same mRNA transcript and read it in a different way by simply shifting down one base. And so if the ribosome recognizes GUC as the first codon, then UUA will be the second, CCG will be the third, CAU will be the fourth, and so on. And that will result in a different polypeptide order. And so for every RNA transcript, there's 3 different reading frames that can result from that. And this helps to create different proteins using the same mRNA transcript. The universal code is a universal code. All of living cells on this planet follow the same rules. And so this table that I've been showing you is the same table followed by every organism on the planet, with some exceptions, actually. Of course, there's always exceptions in biology. So we like to think of the genetic code as universal, but there are a few exceptions to this rule. For example, UGA is a stop codon. Typically, for all organisms, it's a stop codon. But for this bacteria, Mycoplasma capricolum, the UGA actually encodes for something. And so it's a nonsense codon for most of us. But for this particular mycoplasma, the UGA encodes for tryptophan. Mitochondrial DNA also has an alternative genetic code. For example, the human UGA the UGA codon for humans typically encodes for a stop codon, a nonsense codon, which signals to the ribosome to stop making protein. And that's true for nuclear genes. However, in the mitochondria of a human, we actually see an altered genetic code where UGA stop codon actually encodes for tryptophan in the mitochondria of our human bodies. And so this is actually one piece of evidence for the theory that mitochondria in eukaryotic cells began their life as a small bacterial cell that was engulfed by another bigger cell. And rather than that bigger cell destroying the bacteria, it kept the bacteria and it evolved into a mitochondrial organelle. And one of the reasons why we think that is because the genetic code in the mitochondria is really similar to some bacterial genetic codes that follow an alternate genetic code.